Hello students, welcome to the second part. Now we are going to study the various stages of development of anther, means the microsporogenesis, the events which will lead to the formation of microspores or pollen grains. Let us see how the young anther looks like. We take a TS and see how it looks like. It's made up of meristematic cells covered by one epidermal layer. See? Then it is followed by development of on the four sides towards the four ends. What is happening? That a cell gets differentiated. All the four regions. So this cell, which is hypodermal in nature, the nucleus becomes prominent, the cytoplasm becomes dense, and this cell, which gets differentiated from rest of the cells, which are meristematic, means actively dividing. So this cell which becomes prominent, it is called the primary archisporial cell. This multiplies or divides, giving rise to a plate of cells, a layer of cells or a plate of cells. This plate of cell or layer of cells you can see in all the four areas, four corners. This is called primary archisporial layer. This is called primary archisporial layer, and the connective by virtue of which the anther lobes are connected to the filament is called connective. It is here. See now what happens in the next step. We'll just show only one lobe. So what is happening in the one lobe will be happening in all the four lobes, because meanwhile the differentiation of cells is taking place, the differentiation of layers is taking place, the layer after layer is being formed. There is an increase in the girth of in the size of the anther also. And see, look here, since it is now it has taken anticlinally, it has divided, the cells are divided, formed a plate of cells. Why? The anticlinal division, they're increasing this way. And now you'll see that the primary, this layer, the archisporial layer, it divides into two periclinally, parallel to the surface of the cell. So it is dividing periclinally, not anticlinally. Look here. So that's why it's going out, out, out like this. So you'll see here the layer towards the center of the anther is called primary sporogenous layer, and the outer one is called primary parietal layer. Mind it, students, the sporogenous layer does not divide any further, while as the parietal layer divides periclinally. Can you see parallelly located cells here? These periclinal divisions, one after the other, as 4 to 5 layers of these layers are formed. These layers of cells which are parallel to each other, these are called middle wall layers. So now how many layers are there? Outermost, the protective layer, the epidermis. Just below the epidermis is our middle wall layers, 4 to 5, and inner to the innermost layer is primary sporogenous layer. My dear children, what happens? And normal anthers, in normal angiospermic anthers, generally the layer which is towards the sporogenous layer, the layer which is just near which is touching the sporogenous layer, this gets differentiated to elongated cells, tapering cells, and these cells are called tapetum cells, or we call this layer as tapetal layer. Generally only one tapetum is there, and tapetum is of two types, either wandering type, amoeboid type, or glandular or secretory type. Now in some angiosperms, you must be seeing here in this figure two layers of tapetum. Yes, this type of tapetum where you find two layers of tapetum, it is called dimorphic tapetum. One inner layer of tapetum and one outer layer of tapetum. Whatever the thing, it is a nutritive layer. It provides nutrition to the developing cells. What happens actually inside? They have one single nucleus. With the passage of time, what happens? The nucleus undergoes karyokinesis. You know already what is karyokinesis, division of the nuclei. So 1, 2, 4, they multiply, keep on. No cytokinesis takes place. But these nuclei, after formation, they fuse. Sometimes you can see the polarity level of these cells is more. They are polyploids also. So these disintegrate, degenerate at the end, and they are absorbed by the sporogenous layer. Now whatever the thing, the inner tapetum and the outer tapetum, whatever, you will see after some time The primary sporogenous layer, in this, the changes start taking place. The callose deposition starts taking place. You can see the red cells, the primary sporogenous cells, being covered by the callose deposition. What is happening? These cells are undergoing division. Which division they will undergo? Yes, the reduction division or the meiosis. Because the plant body is sporophytic, it is diploid. Gamete should be haploid. So what is the spores should be haploid. So what will happen? You can see inner to this layer what is happening. The cells are dividing. They are ready for division. Now after this what happens? The tapetum is gone, the inner one. The outer tapetum is there. You can see the radially elongated cells. They are providing nourishment to the developing. So you can see the condition of the nuclei. Somewhere the simultaneous type of division is taking place. Somewhere the successive type of division is taking place. I have already explained in the last slide, in the last part 1. Now look here, after some time, these cells only give rise to tetrads. These cells give rise to pollen tetrads because each cell will undergo meiosis, successive or simultaneous type of division, but each will give rise to 4 cells. So we have tetrad formation. There are different types of tetrad formation. So once the tetrads are formed, then after this, the anther matures. When it matures, the pollen grains are separated. Various types of tetrad formation you can see. You have tetrahedral formation, you have isobilateral type of formation, you have decussate type of formation, means two at right angles to each other. A pair of these tetraspores, two in one direction and two at right angles to this, the decussate arrangement. And then decussate arrangement. And then the T-shaped, where you can find the two these spores at the upper end forming a T-shaped structure and also in a linear fashion. After this, they get separated and each one of it gets its own intine. It has got the intine, it will make its own ornamented layer. So depending upon the type of family, depending upon the type of pollen grain, they will have different type of ornamentation. Once they are formed, they are separated, they put pressure, they exert pressure on the layers. The middle, one of the middle wall layers just below the epidermis becomes endothecium. It becomes fibrous, so we also call them as fibrous layers. The tapetum is ultimately absorbed completely. They disorganize and these spores put pressure and the epidermis becomes, it has little elasticity. It can stretch to some extent, then it ruptures, withers away, and the endothecium becomes fibrous. It absorbs moisture and helps in liberation of the pollen grains. So this is called dehiscence of these anthers, and the pollen grains are shed. They have one single nucleus, and these pollen grains formed are also called microspores. They are haploid. These are some of the references. You can see Maheshwari P and Bhojwani and Bhatnagar and Dantu. I use these references. And last but not least, thank you for patient listening. If you liked it, like it, share it, and subscribe it. Thank you.